So it's been shown in past studies that biodiversity and primary productivity of a plant system are correlated. In the past, diversity has been defined in two ways. One being interspecific variation, which if you increase interspecific variation, that just means you're adding more species to a system. And it's been thought that this increase leads to a productivity increase because you're increasing the number of functional groups. Therefore, you're increasing the number of niches that that system can possibly fill. With regards to intraspecific diversity, which is the second type of diversity that's been studied, if you are within one species and you increase the number of genotypes in a system, it's been shown that productivity of that system will increase as well. However, there have not been specific traits that have been defined, just more genotypes equals more productivity. So in this study, we define diversity as differing traits of genotypes within the same species. We chose these traits to be plant sex as well as plant growth rate, which was pre-characterized in an earlier experiment. We also manipulated the presence and absence of herbivores, and we took data on the above and below ground biomass of the plants, as well as mycorrhizal colonization. Which leads me to my main questions, which are, which particular traits underlie intraspecific diversity effects on net primary productivity? And are those effects contingent on other ecological conditions like herbivory? So for this study, we used a plant called Baccharis salicifolia, which is dioecious, meaning the males and females are separate. If you look at these two pictures, they're of the flowers. The top is of the male and the bottom is of the female. They're very easily differentiated to the human eye. We chose 6 of the fastest growing and 6 of the slowest growing genotypes, which gave us 3 genotypes in each of the 4 distinct categories of fast growing male, fast growing female, slow growing male, and slow growing female, respectively. To implement herbivory control, we built 15 cages. If herbivores were not allowed to be present inside the cage, they were periodically sprayed with insecticide. Within the cage, there were 9 planting locations with 2 plants per location. Most of the data analysis was done in pairs. So each pair was examined with regards to either plant sex or plant growth rate, but not both at the same time. They were looked at with regards to either being monocultures or polycultures. An example of this is the first column here. This is an example of the different plant sex treatments. So the top is a male-male monoculture, the middle is a female-female monoculture, and the bottom is a male-female polyculture. The same can be seen for growth rate here with a slow growing monoculture, a fast growing monoculture, and a slow fast growing monoculture. So all possible combinations were represented multiple times within a cage. We characterized the 4 genetic classes, main points being that fast plants grew faster than slow plants, as we expected. Males and females had similar growth rates, and females had more investment in below ground biomass and had higher amounts of mycorrhizal colonization. The interesting effects, if you look at the top, this is showing the slow, fast, and fast pairs. This is their growth rate. If you look at the height of all of these bars when herbivores are added, there's a consistent decrease across the board, showing that herbivory has a strong effect across all possible combinations of growth rate. However, this middle bar shows that there are no diversity effects seen with regards to growth rate. So the slow and the fast lie on either side equally, so that this is just an additive effect of both of the possible genotypes. However, with regards to plant sex, this being a female-female monoculture, this is a male-male monoculture, both of these bars decrease evenly when herbivores are added. However, the polycultures, when herbivores are not present, grow less than is expected of them. When herbivores are present, they grow more than is expected. Our hypothesis for why this happens is that males and females have such high levels of competition when herbivores are absent that the female outcompetes the male, which causes the male to do so poorly, the overall biomass of the pair decreases. When herbivores are present, this releases some of the competitional pressure between the two, and that allows the pair to outperform either the female-female or male-male pairs because the females can allocate more resources from underground. In conclusion, fast-growing genotypes grow faster than slow-growing genotypes. Males and females grow the same. There were no diversity effects seen with regards to growth rate, but we did see them with regards to plant sex. We also discovered that there are potential variables with being able to see these effects. So if we had not excluded herbivores, we may have not understood these diversity effects the way that we do. There are other possible variables that could affect these diversity effects, such as resource availability, like nitrogen and phosphorus in the soil, as well as possible plant stress, like drought. As well as the plant traits that cause these diversity effects may not be what we expect them to be. So a trait that is highly varied, like plant growth rate, that you may expect a slow and fast-growing plant growing together to allocate resources better than a fast, doesn't actually happen. But if you combine plants that have different characteristics that involve a whole suite of traits, like plant sex, diversity effects may end up being seen. I would like to thank my PI, Dr. Keelan Mooney, as well as my program directors, Dr. Marlene de la Cruz and Luis Motabravo. Thank you.